All right. The last edition. So we're going to finally, I've been talking about, in a referential way, I've been talking about exercise genomics for a few lectures, saying this is something we'll get to. It's actually the first lecture I ever gave before I came here was exercise genomics, was the topic. And I, as a throwback to my old lecturing self, I put in 3 of those slides with the little Yukon logo at the bottom and everything. I pulled up that slideshow this morning and took a few slides out of it. So it was in 2012, and I sort of had dropped the subject of exercise genomics since 2012. I haven't really talked about it, but so this is the field is young, and we'll talk about that. The field is young, and it's a little bit inaccurate and overambitious. But it's still worth talking about. So how does our denim, right? How do our genes help determine athleticism? We've talked a lot about the inverse. We've talked a lot about how our physical activity, our exercise, our behaviors influence gene expression. What can we do or do differently to affect the behavior of our genes, translation of proteins, phenotypic traits developing? What can we do to elicit those outcomes? And today we'll talk a little bit more about what genes do to affect athleticism, sort of the determinism of genes. The points haven't changed. Here's what I'm thinking. We'll have one more day where we'll do a competition and so have this in your head as we go through today. You're working with an old person, right? So on the test, there was a kid, and we did Oprah and we did Wonder Woman. Wonder Woman needs to gain muscle. Oprah needs to lose fat. Here's a kid. Like we've done a few different populations, but we haven't done old person, geriatric person. We haven't done that group yet. And so that's something we might do as a group if you're not opposed to it. You know where to find stuff. Just keep these in mind because, again, this is critical, but we're going to start with telomeres. We're just going to refresh telomeres and then get into exercise genomics. So remember the aglet, the little cap on the end of a shoelace. That is your telomere capping the ends of your chromosomes. And it provides a little shield, right? It's a little defense, it's a little shield at the end of your chromosomes. And it lives in the nucleus, and as mitosis happens, as we replicate stuff, we chop down those telomeres. Those telomeres get shorter and shorter and shorter. So in a person, TTA, GGG, TTA, GGG, TTA, GGG, over and over and over, those repeat sequences. And again, how many of these base pairs do we see? 10,000 to 15,000 base pairs, 2 to 50 kilobases, 1,000 pairs, over and over and over. And again, as cells divide, we shorten our telomeres. Our telomeres get shorter and shorter and shorter. We do have telomerase, telomere-based telomerase. We do have this, and it's critical in some cells, right? It's critical in the hair follicles and the lungs and the immune cells and the hippocampus and bones and that's it, skin. I don't know. Skin. What else? In the heart, some smooth muscle. It's critical, but we don't use telomerase as potently as some of Earth's critters. We're not very good at telomerizing ourselves. And so senescence eventually develops, this age-related shortening of the telomeres until you're no longer replicating, until these until these cells no longer have functionality. Now think about functionality from the perspective of like a pancreas. If you have senescent pancreas cells, okay, now we're having insulin problems. Now we're looking at diabetes. Wherever these cells reside, where you get really short telomeres, it's not just that replication is inhibited. Functionality is also compromised. And have you ever like brought home like a bag of bananas or avocados or lemons or apples or oranges or something, and there's like one rotten orange, and like 2 days later, all of them are rotten? Senescent cells have that effect on their surroundings, mostly through inflammatory release. So it's important to try to keep all of our cells healthy for as long as we can. And at every single time, these cell cycles, we're looking at these telomeres shortening somewhere in the range of 30 to 200. Let's narrow it a little more, 50 to 150. These base pairs get shortened on our telomeres. And oxidative stress is a huge culprit. What we would take, vitamins C and vitamin E, what we would take these antioxidants for oxidative stress, it attacks those GGGs a lot. Those GGGs in the telomeres get attacked by free radicals. And so it's helpful to have antioxidants on board, and especially endogenous antioxidants, and chronic inflammation too. So when we look at stuff like metabolic health that is growing compromised, look at metabolic syndrome, and what do you see? Insulin insensitivity. You see a distending belly, right? The abdominal adiposity, and these things, especially abdominal fat, is associated with additional inflammation. And so with an oxidative profile and an inflammatory profile, you start to age quicker at the cellular level, at the level of your telomeres. Now, the health span and the disease span. Health span has been in the physiologists, health practitioners' parlance for a while. The disease span is being talked about a little bit more in recent years, but somewhere in the middle of life, this is like the midlife crisis zone, where people tend to start exhibiting disease risk, if not disease, at least pathological risk, a likelihood that one ailment will arrive and be compounded by another and compounded by another. The disease span, when somebody is 60, 70, 80, 90 years old, they're at a higher likelihood of whatever illness. It sort of doesn't matter what it is. Everything. They're at a higher likelihood of getting those things. Give me your question.
Yeah. For the oxidative stress, so cardio is good for health, but it's also bad for. I thought like cardio is like you want to do cardio for lengthening your lifespan instead of like lifting weights for like mTOR, that's detrimental sort of. But then it's also gives oxidative stress.
That's a really good question. Really good observation. It wasn't a question yet, but I will receive that as a question as phrased, and it's a really good one. So acutely, exercise increases inflammation too. There's an inflammatory response with exercise. There's also, just look at the electron transport chain. This is how we're providing all this ATP during exercise. It's a bunch of oxidative metabolism, and we're going to get oxidative stress from it, the premature leaking of electrons to the oxygen we're breathing. We're going to get superoxide, right? Reactive oxygen species, a free radical with oxygen in it, superoxide. That's endogenous, and we generate a ton of that during the electron transport chain. So during exercise, we get an acute increase, an acute burst of both oxidative stress and inflammation, followed by a chronic suppression. And so think about exercise and your heart rate. You go exercise and your heart goes and goes and goes. You're on the treadmill or whatever. Your heart is going, going, going. And it's acutely increased, but chronically your heart rate, your resting heart rate goes down. So if you are a serial exerciser, the number of heartbeats you experience during the day is lower, despite during exercise. And you see a similar thing with free radicals, with reactive oxygen species, oxidative stress and inflammation, both. Exercise increases acutely followed by a chronic reduction. So it's a really good observation. So the health span, and then at some point in life, if we live long enough, right? If you die at 14, okay, there's tragedy because you have a lot of health span left. But at some point in life, if we live long enough, we cross over into this age-related disease span where we're at risk of experiencing cardiovascular and metabolic and pulmonary, just name it, everything. Cognitive decline, everything starts to arrive in this disease span. And one mechanism, one explanation is the shortening of the telomeres and the rate at which we progress into that disease span. Now, what we don't want, it's fine, this is where the average person crosses, right? What we don't want is to cross there. And there are pathologies, right? Short telomere syndromes, inherited, right? You're born with this. And it is, I don't know, it's not as fascinating as Benjamin Button, but there's a rate of aging that sets in so rapidly. Really rapid aging, where you're going to see like teenagers with age-related wear and tear and gray hair and pulmonary complications and cardiovascular complications. I mean, just the list, the liver is at risk, everything is at risk. And what this is a genetically assigned telomere length that truncates a lifespan. But it doesn't truncate the lifespan like you're healthy, and then, death befalls you. The reaper comes and just swings that scythe at whatever. You transition into that disease span at a really young age. And so that illustrates the importance of telomeres in aging. Telomeres aren't everything, but when you see this STS, short telomere syndromes, right? These short telomere syndromes, you might as well be 60 when you're 20. It depends on the length of telomeres that you're assigned and the rate of their attrition, the rate of their decay. Now, what you do want is to drag that health span way into your later years. You want to be 70 years old and say things like, I got the back of a 20-year-old. I got the colon of a 25-year-old or whatever. Whatever it is that is a marker for health. I love Bill Murray, but he's like not that old. When look at pictures of Bill Murray at 40, and he looks like he's like 106. And wonderful actor, Groundhog Day and Ghostbusters and whatever. And you can find like YouTube videos of him just like crashing college parties and stuff. So wonderful guy. I just happened to put him here, not as a way of mocking him or anything, but just as a way of showing that Bill Murray and Sandra Bullock, right, they're not aging at the same rate in terms of apparent kind of outward appearance. George Clooney is not aging at the same rate as Bill Murray. Just look at when they're the same age, and they look nothing like each other in terms of that trajectory. And the trajectory that you see on the outside, if you have senescent cells that are visible to the naked eye, the invisible ones are likely to be similar in their telomere length. The cells that you can't see, the cells in your hippocampus and in your liver and in these cells throughout the body are likely to resemble the visible cells that are showing signs of age-related wear. I could put 1,000 people over on the like I'm aging badly section, like the Rolling Stones, the entire band. They looked like this when they were like 25. And so there's a lot of people we could put over there. There's like whatever, it's more people. Rachel from Friends, what's her name? Jennifer Aniston. She could go over there, I think, right? Yeah. So we all know people who age well and people who don't age well. And it doesn't have to be actors, right? We know people in life. And part of that explanation is going to be these truncated telomeres, the odometer that's just winding down, right? It's a backward odometer. Your life has a certain number of miles on it, and your odometer starts at full capacity in the mileage. And with every mile you cover, that odometer winds down to 0. Now this, the Guinness Book of World Records, I just pulled these up yesterday. So the oldest astronaut male, not astronaut, but the oldest person to go to space, the address just happens to be oldest astronaut, William Shatner, right? So in October 2021, he was just over 90 years old, and he looked pretty good. He went up into outer space. The oldest person ever lived in France. She was just over 122 years old when she died. And apparently, if you listen to her story, like she took up fencing when she was like in her 80s, and it was, I think, her 100th birthday. She like walked around, and like everyone was wishing her a happy birthday. I mean, she has this, you read about her, she has this real story about her life and her physical activity. It's possible for a human genome, not every human genome, to have lives like these, and to extend into that health span for a really long time. There are certain behaviors, there are controllable behaviors that we can do. And we talked about some of those in the last lecture. I just pulled up a lot of the literature in the last lecture, and we went through stuff like smoking and environmental exposures, for like BPAs and pesticides, coffee, right? So we went through a lot of those things in the last one. And then I decided I'm just gonna reread this book and create a little like book report for it. It's really good. So I reread the whole thing yesterday. And I took notes on every single piece of advice that was worthwhile. Remember, Nobel laureate. This isn't just like a pop fiction-y kind of, this isn't like Dave Osprey from Bulletproof writing his blog or whatever. This is a Nobel laureate whose work on telomeres is what earned her Nobel Prize. And so she went through the literature, and everything that had support, some amount of support in the literature, these two authors, put together in this book. And so I went through the entire thing again yesterday, and just wrote down everything. Now, some of this is employable, right? Some of this is you can prescribe to people or use with yourself. Some of it is flimsy evidence. Some of it is really strong evidence. But I've categorized it into these different domains: nutrition, environmental exposure, living spaces, living environment, relationships. I've categorized it in different ones. And the first one is thinking patterns. Right now, people who ruminate, I do it, everyone does it. Unless you're super mentally healthy, you do it too. We have like thousands of thoughts every day. Who knows how many? You see these estimations like we have 70,000 thoughts or 65. Who cares? That's not a real number. But we have a lot of thoughts every day, and a lot of those thoughts are just repeats. I don't know, 90% or something. These thoughts are like you've had them before, and you're just clinging on to something that you thought before, ruminating about it, just marinating those thoughts in bitterness and whatever. Don't do that, right? That appears to be bad for telomeres, right? These negative thought patterns. Now, thought awareness, think like meditative thought awareness, where you're very reflective of your thinking patterns. That seems to be good for telomeres. Angry, hostile reactivity, right? You're in the I don't know, someone like cuts you off and you get road rage, right? That kind of reactivity, that's bad for telomeres. To have anything where you feel like violently slighted in whatever the circumstance is, that's bad for telomeres, right? Everyone, like on the road, everyone's just trying to get to where they're going. They accidentally got in your way a little bit. Like, okay, now we have to honk and scream and stuff. That's bad for us. If there is a winner and a loser in that correspondence, if there's a winner and a loser in that relationship of road rage, the person whose heart rate does this, whose blood pressure just squeezes, right? The person who starts to perspire, while flipping people up, that's the person who lost. That's the person who lost that exchange because their telomeres are shortening, among other health complications. Self-tolerance, self-compassion, those also seem to be good. And this goes along with the ruminating part. Now, a lot of these are from psychological inventories, questionnaires, where they ask people a bunch of questions and people self-report their psychological state, their moods, their sort of mental state. It gets a little bit flimsy in terms of, you're not measuring pH, right? You're not measuring something that's objective. What you're measuring is people trying to be objective about inherently subjective criteria. Given those limitations, being like self-compassion, not kicking yourself over everything. So like, oh, I fucked up a little, that's okay. Next, I'll get another round at this. But pessimism, optimism is good, pessimism is bad, but pessimism is worse than optimism is good. Negative traits seem to be more destructive to our telomeres than positive traits are helpful. So the bad stuff tends to affect us at a magnitude greater than the good stuff. The good stuff we should employ, but the bad stuff is better to avoid than the good stuff is to employ, if that makes sense. One of the biggest things that is controllable is about how we see the coming day, the coming events. If finals are coming up and you just experience dread, panic, worry, dread, you think you're gonna fail, you're just, I can't bear to do whatever. Terrible for your telomeres. As opposed to, elite athletes, they want to be called in when there's like 6 seconds on the clock. There's like 6 seconds on the clock, they're down by 2, let's just say it's basketball. Soccer, you're down by 2 with 6 seconds. I don't know what to tell you. So let's just say it's basketball. And you're down by 1 or 2, there's 6 seconds to go, it's a high pressure. There are people who are excited about the challenge. They can't wait to try that out. And it could be to have that reaction about anything, to have that reaction about exams, to have that reaction about, let's say whatever you're doing for work. Let's say everyone in here becomes a professor. And to have that reaction about class, to have that reaction about whatever lies ahead of you. To see it as a challenge and encounter it with excitement. You're supposed to have epinephrine. You're supposed to have cortisol in these situations. That's supposed to mobilize energy substrates. That's supposed to prepare you to overcome that challenge. But if you dread it, you're going to have this lingering cortisol response, shortening telomeres, not helpful. And then there's flimsy evidence for having a purpose in life. But it's like when people retire, you see the downfall. You see this slide into a lack of capacity and sort of a lack of that beaming, kind of jubilant personality that people used to have. They just sort of stare at you, after retirement. Having a purpose and pursuing that purpose seems to be helpful in a lot of ways, and maybe it's helpful for telomeres. So those are thinking patterns. General stress, we can't avoid if we had childhood trauma. That's not something that we can, put on your time turner, like the Harry Potter time turner thing, and reverse. You can't do that. So you just have to accept that if there was repeated neglect or abuse or some sort of childhood trauma, that leaves a record in our telomere receipts, right? We have a record of that payment kind of put upon us as children. That's hard to cope with because we don't have control over that. But for everything we have control over, short-term stress, totally fine. Finals week or some difficult athletic thing or a work situation that doesn't last all that long, whatever it is, that's fine. Short-term stress is totally fine. And so you remember when we talked about the Baltimore, Maryland, the shipyard workers study and stress. And stress bolsters our health. Manageable doses of stress bolster our health. Huge doses of stress break us down. And so finding that little sweet space, our kind of Goldilocks encounters with stress. But a life devoid of stress and coping is terrible for a lot of reasons. And that's sort of what America is becoming, this sort of cradle of no stress. And that's what makes that so challenging with our safe spaces and stuff like that. What makes this so challenging is people don't develop the coping skills. And so then when life inevitably beats them up at some point in life, not physically, but figuratively beats them up, we don't cope, right? And our inappropriate coping leads to exaggerated cortisol responses and shortened telomeres.
When you say stress, you mean mental stress?
So I'm talking about, in this case, really good point. General stress, mental stress right now. We'll talk about exercise in a little bit. But this would be the same in terms of the profile with physical stresses. So if you think here's, no stress, terrible, whether it's physical, emotional, mental, psychological, whatever it is, too much, terrible. We want to be in there with all stresses, but right now I am talking about sort of emotional, psychological stresses like that. So we need some stress. If we're just like, oh, the whole world is nice to me all the time, we're going to wind up with shortened telomeres because you're going to encounter a point where the world is unkind. Okay. Humanity, we're not that nice of a species when you encounter enough people, when you encounter enough of us. Exaggerated stress response. That's probably the worst thing in terms of the literature. And we talked about this one, the exaggerated stress response. I pulled up a study in the last lecture. And if you have, two people could encounter the same stimulus, and one person has this big exaggerated response, that kind of hypothalamic pituitary adrenal exaggeration associates pretty strongly with shorter telomeres. Good management of stresses is good, right? There are ways to manage stress, healthful ways to manage stress, and you see those corresponding to longer telomeres. Unmanaged stress and like bad relationships, bad neighbors, a terrible work environment, unmanaged stress. Short term, that's okay. Long term is bad. Anxiety and depression. Depression is harder on our telomeres than anxiety. Generalized anxiety over a long period of time is hard on the telomeres. Generalized anxiety that's very severe in nature is hard on the telomeres. But if you're looking back 10 years ago at a period of exaggerated anxiety, it doesn't leave that much of a paper trail. It doesn't shorten the telomeres in a really recognizable way. There's an acute shortening of telomeres in the presence of anxiety, not like you're a little bit anxious about the event to come, but sort of a generalized anxiety, I better get on some meds kind of anxiety. But depression, if these events turn over into depression, that's when you start to see a little bit more severe telomere shortening. Depression is harder on our telomeres than is anxiety, and in particular the hippocampus. We're going to talk more about the hippocampus starting on Friday when we transition to exercise and cognitive health and finding the zone and stuff like that. And in some depression cases, you do see elevated telomerase activity, but it's probably a compensatory mechanism. It's looking at these telomeres that are shortening, sort of erasing their way into attrition, and the compensatory mechanism is not particularly effective. So you do see elevated telomerase in some patients with depression, but you also see shortened telomeres.
If you take like medication or something that takes away or like lowers the ability of anxiety and depression, is that like, does the medication affect the like the symptoms of like depression, or is that like it gets rid of a substance that is causing like the telomere?
That's a super complicated question. It was phrased in a clear, direct way, and it's a super complicated question to ask. So relative to telomeres, you see often with medication more telomerase activity. Following that, you do see some symptom management. Where it gets really complicated is in the nature of publication, not the journal Nature. Although this might have come out in Nature. I think it was New England Journal of Medicine, actually, when this one came out, which drug trials have to register the actual study. And that doesn't mean they have to publish their study. That has to be registered. And somebody came along and pulled up all of the SSRI studies that were unpublished. Now, what pharmaceutical companies can do is they can start like 20 trials, and then they can stop trials whenever. Oh, we're currently showing an effect. Let's halt the trial now, as opposed to let's see it through till the end. They can do 20 trials and find a positive effect in 6 of them, publish those 6, leave the others unpublished. So now it looks like every single study that's ever been done shows a positive effect, thus SSRIs are effective. And somebody went and dug up all these unpublished works, and it's like a 50-50, it's like a coin flip whether these drugs actually work. Now, in the published literature, it's not the case. They look very effective. But in terms of the actual studies that were conducted, flip a coin, of whether the study is going to turn out effective or ineffective. Not necessarily bad adverse outcomes, but ineffective. So it becomes a little bit harder in pharmaceutical research to tease out the right findings. That's why I say it's a really complicated question, and I didn't answer it well, but I hope that gives you an illustration of why it's tough to answer. But I wouldn't ask a question like that. It's just a fascinating observation that you see. Sleep, it's like the most important thing in life. The pillars of health. The pillars of health are sleep, exercise, physical activity, exercise, and nutrition. If one of those pillars is missing, you're gonna have a hard time being upright. You're gonna have a hard time preserving health if you're missing a pillar. When you miss a pillar, you can prop it up with medication. Whatever it is, if it's bad sleep, if it's bad nutrition, if it's bad exercise, there are medications that can serve as a crutch. But you don't want to rely on medications for something that those pillars should be doing, should be supporting you, right? They should be providing those load-bearing beams for your health. Should be exercise, physical activity, exercise, nutrition, and sleep. What people eagerly sacrifice is sleep. And man, you shouldn't do that. It's the most important one. It's the most important one. And you see it in the telomeres. In stress physiology, I think I did 5 lectures straight on sleep quality, sleep health, consequences of bad sleep, strategies for optimizing sleep. But for now, if you're getting at least 7 hours, good quality sleep, a regular sleep schedule, your telomeres thank you for it. If you're getting fewer than 5 hours, or you have a schedule where your work schedule, Nathaniel's work schedule. I was thinking about you, Nathaniel, when I made these slides, your work schedule. You can tolerate this for a while, but for too long, it starts to take a toll. If you do it for too long, it starts to take a toll on your telomeres. Sleep apnea or closed off breathing is bad. And then toggling your sleep schedule is also bad. If you can have a good sleep schedule, really good for telomeres. Behaviors, everyone multitasks, and it turns out the evidence indicates that that's not good for telomeres. Unitasking, that's what we should be doing. And I'm gonna move into this idea of what flow is chemically, the neurochemical cocktail for flow or the zone, right? We're gonna get there soon. And but if you can find this unifocused task where you're actually mentally present with what you're doing, you're emotionally and psychologically engaged in the task, that seems to be a lot better for a lot of things, for mental health, but for telomeres as well. Meditation seems to work in a number of studies. Mindfulness meditation and other forms of just of being, conscious of your thinking, being mindful of your thoughts, paying attention to the moment, that stuff. So meditation works. That wandering, uncontrolled mind seems to be bad. People who are just sort of lost in thought and have, 50,000 thoughts a day that are distractions, and you pursue those distractions, and you're just sort of, whatever thought comes to you, you grab it, and you're like, that doesn't seem to be good for telomeres. Conscientiousness, so having a duty in life, having work that you connect with, having a purpose, and taking that purpose seriously, that conscientiousness seems to be good. Impulse control, delayed gratification, stuff like that correlates pretty strongly, but again, correlates. Up here, I say numerous lifestyle factors exhibit correlations with telomere length. Some of these are stronger than others. But again, when you get into conscientiousness, we're talking like, psychological inventories. It gets a little bit flimsy, but the correlations indicate. Social environment, we know that poverty is bad for telomeres. Above poverty, it doesn't really matter if you have, if you're like Jeff Bezos or like a professor, where it's like, well, I'm not struggling for $1. So the extremes of wealth don't really seem to matter above poverty. Once you climb out of poverty seems to be bad. Everything else seems to be okay. Now, wealth does correlate with a lot of behaviors, access to healthier foods, access to a nicer neighborhood, stuff like that. But if you control for those factors, only poverty really seems to be bad. Social isolation, bad. Right? You have to have relationships. You have to have connectedness with people. Good relationships, wonderful. Good relationships are really helpful. And understanding of each other and providing support. Bad relationships where there's chronic stress, that seems to be bad for telomeres. Discrimination. Now they made a point of saying when it's internalized in the book, which applies to everything. They made a point of saying that with discrimination, whether it's sexual orientation, whether it's race, sexual discrimination, whatever it is, it doesn't seem to affect anybody unless it's internalized. But that same thing would apply to every other one of these stressors, these social stressors. You can't not internalize physical stress. If you get like hit by a bullet, like, okay, well, I'm not going to internalize the bullet, right? I'll just like bounce off. But to internalize psychological social stresses, that seems to be critical in terms of the telomere relationship. And so they did make a point with the discrimination one. The living environment where you are, neighborhoods with low social cohesion, bad. If you don't know any of your neighbors and everybody is like, it's just strangers and you're suspicious of everybody, and that doesn't seem to be as helpful as a community if you live in a community. I know wherever Nathaniel lives, people smile at each other. Because I see Nathaniel do it. I see Nathaniel smiling and waving at the neighbors and acknowledging them. And so that sort of social cohesion is helpful. Stockton et al. Okay, Stockton and other neighborhoods in which you feel unsafe. We're going to talk more about the amygdala soon. The hippocampus and the amygdala. Some of these brain areas are coming soon. And if you're on high alert, if you're vigilant, if you feel endangered all the time, that's bad, right? That's bad for telomeres. Green, lush and leafy environments, right? If you're in your park spaces, if you have, you look out of your window and there's just plants everywhere, that seems to be good, right? As compared to like San Francisco, they specifically mentioned, I think live in San Francisco, because in the book, they talked about going on walks while writing the book through San Francisco. And so they reference like San Francisco, where there's just tons of litter and no green anything, is how they described San Francisco. That was me paraphrasing their description of San Francisco. But that seems to be bad. That seems to be bad for telomeres. So if there's no green spaces and like litter and you're on high alert and there's no social cohesion, that's bad for telomeres. A living space where you know your neighbors, you don't feel threatened, and there's green stuff, that seems to be better. Environmental exposures, we talked about some of these. Just in general, pollution and chemical exposure is bad. And everyone sort of knows that, right? We talk about cigarettes, we talked about pesticides, they talk about mechanic shop chemicals, they talk about dyes and cleaners, carbon monoxide, cadmium, lead, PAH, BPAs, we talked about BPAs, but other industrial chemicals that just aren't as famous as BPA yet. Canned foods, it's like, I don't know, 10% of them, something like that, still have a lot of BPA in them. But just if you eliminate BPAs, there's other industrial chemicals that take their place. And we just haven't done enough research on those things yet to identify the health consequences. BPA, there's a lot of research that's been done on that in a number of directions. Only one of those directions is telomeres, a lot of other health complications associated with it. Scented products, some of them are bad. I don't know how to decipher. So, smoke your candles if you want. Whatever. Your perfumes, your candles, sometimes they're bad, but I haven't seen the actual studies on those. That was strictly what they said. Excesses of UV exposure. If you have, just see, you can see those. I don't know if these things are real. I've seen the pictures before. It's like truck drivers. Have you guys seen these pictures where it's like half of their face is just aged? I mean, they look like an old wizard or something. And then the other half is like a kid. It's like the window side. They hit all the sun. And I don't know how Photoshop those things are, or whatever, but it's an illustration. Whether it's true doesn't matter. It illustrates that UV exposure has aging effects, and part of those aging effects are with the telomeres. And arsenic is fascinating that it can actually elongate telomeres, but it'll give you cancer. It'll elongate the telomeres. So what's something that Kevin asked yesterday, or whatever, Monday, whatever it was, about the sort of juxtaposition of these telomeres and cancer. And so arsenic, although it's carcinogenic, it does seem to have an effect of elongating telomeres. Maybe there's a pharmacological intervention here in the future that's really well controlled. Nutrition. Now nutrition can be hard to make valid claims on, because it's all epidemiology for all of these big associations. And when you see like meat causes cancer, or whatever it is, these sorts of claims, the type of people who were eating that much meat were also doing other things that might be carcinogenic in nature. Or maybe their sleep hygiene isn't very good, something like that. So it gets a little bit hard. Coffee and green tea, both of them are good. Remember, there was that study that I had up that talks about caffeine differently from coffee. But coffee and green tea, both of those are good for telomeres. A heavy alcohol load is bad, but like a light boozing, that's okay. There seems to be no relationship with telomeres until you get to heavy drinking. Nuts and seeds, we talked about that. Those are good. The worst thing you can the worst thing you can do nutritionally is high sugar, high refined carbohydrate, high glycemic effect. Eating those things, the sugary beverages stuff, you have like Mountain Dew or whatever, are truly horrible for your telomeres. I mean, among the worst things you can do, other than, I don't know, maybe like smoking or something, but really bad on this for your telomeres. Seaweed is good. Fatty, like omega-3 sources, like fatty non-farm fish and other omega-3s. This one, I read this study. I decided not to put it in. I'm the one that talks about hot dogs. I decided not to put that study in when I'm preparing a lecture. I'll download like 40 studies, and I'll put in like 20 of them, because I'll decide about 20 of them aren't good enough to make the lecture, to make the little file folder. And this was one of them that I decided wasn't good enough. They talked about it in the book. But what they did, they looked at too many foods for my statistical taste. And if you're looking at correlations and you pull up like 50 foods, you're going to find correlations. And so highly processed meats, right? You got like the Oscar Mayer stuff, and but hot dogs specifically emerged as the worst ones for your telomeres. But I'm not really sold on that study, owing to the methods of how it was done. Maybe in the future, more studies will confirm that finding, and I will be happy with it. But right now, it's okay to avoid highly processed meats. It's advisable, because highly processed meats have a lot of correlations that are bad. This one, I've only seen it once, and it was in a study that looked at a lot of correlations. Like there are studies that come out that say like, particular breakfast cereals make it you're pregnant. What should you eat if you want to have a boy, or what should you eat if you want to have a girl? Breakfast cereals correspond to having a boy. Like, okay, but if you assess 600 different foods, correlations emerge. So it's just random chance that you're going to see this stuff. I think that one specifically came out in the teaching journal. The breakfast cereal begets a boy. I think that was in the teaching journal that was talking about the abuse of statistics. But more on nutrition. Charred meat, that's bad. We talked about the dairy fat. A lot of dairy fat, high milk fat seems to be bad. A lot of non-fish animal fat. Fish fat is good. Fatty fish is good. Non-fish animal fat doesn't seem to be good. Dietary sources of just antioxidants. Remember, these antioxidants are helping to, now if you're taking it in pill form, a lot of pills don't seem to be as effective as getting natural sources of these things. But the ratio of vitamin C and E to these isoprosteins, prostaglandins, right? You know what prostaglandins are? Made from arachidonic acid. If you oxidize those things, you can get isoprosteins, inflammatory, whatever marker. So the ratio of C and E to isoprosteins has been shown in the literature as being good. Vitamin D may be good. Maybe it helps with telomerase. And the flavonoids are just the stuff that gives fruit its color. Fruit and vegetables, the stuff that makes it all orange, the stuff that makes it purple and blue and red, the stuff that makes it those colors, these polyphenols, healthy, good for your telomeres. Good for a lot of things, but good for your telomeres. And then you can't avoid pesticides. It can't be done because it lives on in the food chain. Every organic thing enters your mouth, whatever, and it's in the food chain. There's just so much pesticide, and all of the pesticides, nearly all of the pesticides have associations with shortened telomeres, among other complications. But we're working on in the direction of organic. And then for exercise, cardiovascular is best. There are studies on resistance training that aren't compelling for telomeres. Other components of cell health, other components of preservation of cell aging, preservation of cell health, resistance training is good for. Cardiovascular activity so far in the literature seem to hit the high intensity interval training, but enhancing aerobic fitness, exercise that does that, and just having aerobic fitness, but performing exercise that bolsters, that augments aerobic fitness is helpful. A lot of exercise isn't really much more helpful than getting some exercise for telomeres. Too much exercise is bad. Overtraining syndrome associates with shortened telomeres. But anything shy of overtraining, so if you just get like, oh, I'm pretty physically active, I'm getting in a fair amount of cardiovascular exercise, and here's overtraining, and here's where I am, anything in there, you're about the same in terms of the effect on telomeres, says the state of the literature. Now, remember, the type of person who, because we're not doing, at this point, we're not doing a bunch of randomized controlled trials and manipulating some particular variable and looking and then measuring, pre and post telomeres. You don't have to do serial tests for telomeres. You can do your telomere testing. Just Google telomere testing. Some of it's under $100. I think all of it's pretty much under $500 if you want to know the length of your telomeres. But you would want, I don't know that I would trust most of them. Most of these companies, I don't know that I would trust. And you'd want to do serial testing and look at the differences of these because not every single cell is the same. But with the telomere associations, just keeping in mind the type of person who does cardiovascular exercise might be the type of person who eats more broccoli. The type of person who eats more broccoli might be the type of person who has better sleep hygiene. The type of person who has good sleep hygiene is probably someone who doesn't smoke. So the type of person who behaviors and traits move in clusters. It's hard to isolate an individual effect in epidemiological surveillance of this nature. You can control for some stuff. Regression modeling, you can hold some stuff constant. You can hold smoking constant. And it's a little bit harder to hold exercise constant because you're relying on people to say, well, how much exercise did you do? Oh, yeah, quite a bit. Okay, let's mark you at a 6. So it's hard to control some of these things. It's hard to control nutrition. So a lot of these variables, how much do you sleep at night? People don't really know, unless they've been to, like been in a sleep study or had an overnight at one of those labs, then they don't really know. Now, here's something, if all of those were bad for you, if every single one of those was, I have no telomeres left. My cells are done replicating. That's it for me. Like if that's how you feel, because that list just described all negative things about you, here's one that's encouraging for everyone in the room. Education. Now, again, the type of person who does education, right? But having additional education associates with longer telomeres. Now, when they separated, in one of the studies looked at white and black, and it was especially pronounced for black. But there is a relationship with education level and length of telomeres. And I put both of these studies in the file folder. You'll see it later today. I put all 4 of these studies in the file folder. This is just saying exercise works. Exercise works. And this one, they found a lengthening of the telomeres in obese women. And so in particular populations, the effects on telomeres may be more or less pronounced. But again, aerobic conditioning seems to be the best kind. But you can say this is 2020, 2021, 2019, 2018. New studies. This isn't something where you're finding evidence in 1982, right? This is new work. So there's a lot left to discover in telomeres and their relationship with our health and our behaviors and how they influence telomeres. Just remember, telomeres aren't everything. Telomeres are important, but they're not everything. There are other hallmarks of aging. Telomere attrition, that's a big one, that's a huge one. If you have a short telomere syndrome, you're going to see like, oh, is this guy like 19? You're gonna see this accelerated, this advanced aging if you have advanced telomere attrition, and it manifests all over the place. I mean, you get gray hair as in youth, and so telomere attrition is a very important one. That's not everything, but there are other components of aging that are important. I see people writing frantically. I'll leave the slide up for a few more seconds. I will also have this slide. I'm going to put the review slides up soon. Maybe tonight. I'll put up the so far review slides, and this slide will be in there. But yeah, keep writing as fast as those fingers will go. For now, I'm still hearing keystrokes. Okay, now I'm switching it. Now, I'm not switching it yet. So going back to all of these, we've been talking about telomeres, but the behaviors that affect telomeres, whether it's nutrition, whether it's exercise, whether it's sleep, whether it's environmental exposures, whether it's social health, all of this stuff is going to affect a lot of things. It's not just going to affect telomeres. And so when you start seeing some evidence like this, looking at ketone bodies and aging, they talk about telomeres, ketogenic diet, or just eating ketones, and associations with aging. There's a lot left to learn about how diet influences aging. There's a lot left to learn. Some of the emerging data is very compelling about ketogenic diets. I'm not saying everyone should be on one. I am. I'm not saying anyone else should be. But in terms of the aging, there is some compelling evidence in a number of domains. Now, you remember with older adults begins with age 50 if they're shortened telomeres, right? If their biological age is older than that, that's fine to count a 50 to 64-year-old as an older adult, right? This is the ACSM, 11th edition, the one that just came out. Over age 65, classically regarded as older adults, but in the presence of particular conditions, it's fine to count younger adults as older adults. Remember, you're going to want to know balance, coordination, mobility, physical functioning in an older adult. You don't just want to do chapter 2. You don't just want to do risk classes in older adults. You also want to know their general functional fitness, their strength and mobility and fall risk. And fall risk is a huge topic, fall risk, the predictors of fall risk, how do you evaluate it, how accurate are those predictors, huge topic, but it's really useful to know, because if somebody breaks their hip, it depends how old they are, there goes quality of life, probably permanently, not always. Again, depends on age, and it depends on how ambitious they are about their recovery. But then the exercise prescription, I'll upload this so you can see it, but the exercise prescription, you can also take into account all that telomere stuff, right? So if you have a case study on older adults, if I say, here's an older adult, here's Bill Murray, let's say, and write up an exercise prescription for Bill Murray, you're not just going to want to write, copy and paste the ACSM table. You're also going to want to consider some of the telomere stuff, some of the other relationships, whether it's sleep or diet or exposures to environmental stuff. Consider incorporating that. Say, well, why don't we try to avoid this thing? Yeah, you've been smoking since Groundhog Day or whatever. Why don't we try to back off on that? And in its place, you can have this in your exercise prescription for older adults. Okay, are we set on telomeres? So we'll do 15 minutes, something like that, on exercise genomics. Now, we're not going to talk about changing genetic expression, mTOR or stuff like that. We've had a lot of time on that. We're going to talk about genes as predictive of athletic performance, genes as predictive, like a talent identification program where you send in a cheek swab and they say, you should throw the javelin. You send in your roommate's cheek swab and they say, you should be a water polo player. That's what Cameron's cheek swab said, probably. So this is exercise genomics. And Claude Bouchard, geneticist, we've talked about him before. He's the one who did that study that showed, remember those 4 domains where exercise can worsen them in that study, which is triglycerides, blood pressure, insulin, and HDLs. Looking at these domains of health where there are responders, non-responders, and adverse responders, negative responders. That was Claude Bouchard's work. Now this is from 2015, and he's talking about exercise genomics. And we went through, SEPA, and we went through all of these, you guys all did, the Proteus syndrome, and we went through all of these genetic predispositions for a condition. There's an allele here, and that gene, and if you have that allele, here's what follows, right? Now, exercise genomics, let's take that same concept, the same principle. You have that allele, right? Instead of causing SEPA, right? Now, this makes you a great basketball player because it's that allele and that gene. Exercise genomics. Now, in 2015, a paradigm shift is needed. It had already gotten a little bit out of hand. Exercise genomics had. It had gotten a little bit out of hand. 2015, it was already a little silly. Human genomic features of interest. With the availability of the almost complete human genome sequence in 2001, one could begin to look for deviations from the reference sequence and explore in-depth inter-individual differences at the DNA level. He goes on to talk about how many possible areas, sites for deviations there are. There are about 40 million polymorphic DNA sites in the genome of population studied to date. And I'm looking at these, just millions of possible sites for deviations that you'd have to look at. It's not the BRCA1 and 2 genes, it's the leptin gene. It's not these things. There are a ton of sites. Now, same journal in 2020, 5 years after Claude Bouchard's. 5 years later, they talk about, and remember Claude Bouchard's was a paradigm shift is needed, 2015. Here's 2020. And therefore, a paradigm shift in sports genomics is urgently needed. It's no longer merely needed. It is urgently needed now because of the abuse of all of this. Now, these are companies, we all know like 23andMe. Okay, that's one of these organizations, but there's tons of these organizations that are collecting, the cohort size of at least 100,000 here is 400,000 genotypes, goal for 1 million, over 500,000 genotypes, 176,000 genotypes, about 100,000 genotypes, 225,000 genotypes. I mean, you just go through all how many genotypes, how many people have been genotyped. And there's this direct to consumer products where it says, here's who you're related to. Oh, you're related to Dick Cheney or whatever. Here's your risk for some particular outcome. And there's a lot of them that say, here's your athletic profile. You should be an endurance athlete. Here's your athletic profile. Oh, go be a jumper or a sprinter. So there's these as well. So it's direct to consumer. At present, the vast majority of company claims are more in line with future aspiration and promise rather than current evidence-based reality. For exercise genomics, we're not there yet. We're definitely not there. So here's talking about 23andMe. The next few slides are all going to be giant blocks of prose from that exact same 2020 urgently needed article. About 5 million consumers have done 23andMe. And what 23andMe does is they'll also do internet surveys. And in those internet surveys, they'll do these allele associations. And they'll try to do research. That way, they will also collaborate with universities and then sell data to pharmaceutical industries to try to develop new drugs. Now, I'm not opposed to any of that. I'm not opposed to the sharing of data. I think it's helpful. Pharmaceutical industry gets this really bad reputation for a lot of deserved reasons, but we also need the development of drugs, or we have to develop drugs. And you heard Travis Stiles say how expensive this stuff is to do it right. Travis is doing it right. Not everyone does it right, but it costs billions of dollars with a B to bring a drug to the market. Billions of dollars to bring a drug to the market. They have to make their money back. Otherwise, we'd never have a new drug. They have to get their money back. And so if there are ways to advance understanding by sending your 23andMe data over to pharmaceutical industries, I'm not opposed to that, but it should be known, right? As long as people know that. Now, it is performance, human performance, athleticism, expressions of strength and aerobic capacity. These are really complicated. And so people are looking for, again, talent identification systems to figure out which genes, which alleles, which versions of genes do you have? And can we tailor exercise prescription? Can we tailor, can we tell our kids, our 9-year-old kid, don't waste your time with, basketball, do badminton. That's where you're going to thrive, or fencing, or whatever it is. Can we do that? The answer is no at this point. But human athletic performance has long been assumed to be a polygenic phenomenon, except there are some cases, like myostatin knockouts, the Belgian blues, whatever. Okay, that's one gene, and those cows are the size of a house with no fat on them. Okay, so that's a single gene. There's another example of the EPO receptor, right? Erythropoietin, the EPO receptor. I'm like, okay, sure, you're going to be an Olympic cross-country gold medalist, gold medalist. Okay, and that's one gene. But in almost everybody, in almost every circumstance, in almost every gene, it's a constellation that matters. One star doesn't shine. You cannot fill up your solar panels with oh, the North Star is really bright on my solar panel. I can power my house now. You need a whole constellation up there. And so this, looking for this phenotypic variance of complex traits, you're going to need at least 1,000 people, according to this study. But that's a figure that's reasonable. The number of subjects that you need to test for exercise genomics, you're going to need at least 1,000 of these people. And you can't just give them a questionnaire and say, how fit are you? Are you athletic on a scale of 1 to 10? Because your outcome variable, your dependent variable is meaningless. And so you're going to need legitimate testing equipment. You know, put on the GPS and watch people play games, right, for soccer here. Get a VO2 max test. Figure out what a real dependent variable is, and then get their genome also. And then now, with 1,000 of those people, man, that gets expensive. Go do genome sequencing on 1,000 people and go do a VO2 max and that starts to get really expensive. And so it talks about these large scale cohorts, well phenotyped with good dependent variables. There's a substantial budget. And we have a research grant famine at the moment. Travis talked about that. One of his grants was like cut in half. And so it's hard to conduct these things. And you need multiple studies to confirm what has already happened. That's a problem in this literature, is someone will come out with a finding and then it's not replicated. That first finding may never be replicated. And you need a lot of studies before you have any idea the truth, right, or the reliability of those claims. And the world-class athletes, there aren't that many of them. If you're going to look at what really associates with hockey, okay, like you got to get the NHL players. Okay, you have like Gretzky and like a few other people whose names I don't know. And he retired like 50 years ago. But you have to go get like the stars. You have to get like real people and test them. There's not that many of them, and of that many, not that many have been tested, right? So you can't really do these studies all that effectively. And then just consider something like VO2 max and VO2 with the lactate threshold. Different genes are going to contribute to both of those. Economy of movement, different genes are going to contribute to those. Your aerobic performance depends on a lot of variables. A lot of variables determine aerobic performance, and a lot of genes are contributing to each one of those. So a single SNP is unlikely to have anything to say about athleticism worth hearing, right? So if you do your cheek swab and send it into Atlas Labs or whatever, it's unlikely to give you information that's of much value. Now this is from 2012. This is one of my slides. Garrett Ash and I did a couple of presentations. This was at Central Connecticut State University is where we went and gave this. But here's my little Yukon slides. So this is a screenshot I took this morning of my old Yukon slides. Now this one was talking about the ACE gene, angiotensin converting enzyme, and superhuman, what gives elite athletes the edge? And these articles, right, sort of newsy kind of pop media articles, as soon as a study finds an association with something, what immediately follows is media. And the media usually gets it wrong. So this one disagrees, right? So the angiotensin converting enzyme ACE, there's no association between that and Kenyans, because endurance athletes, that's what this one was looking at, was endurance athletes. And Kenyans, Ethiopians and Kenyans in the Horn of Africa, that is where endurance athletes are sort of the most elite in the world. That's where we're seeing a lot of, where it is common for people to be elite marathon runners. And this is Garrett right here wearing his I Love Ethiopia. He actually owned property in Ethiopia. We did our PhDs together, and this is one of his studies, ASH right there, G ASH, that no association between ACE and endurance athletes and Ethiopians. And he has this, I don't know if he still has it, running across borders, his company, and he would train Ethiopians to come and do like the Boston Marathon and stuff. His first ever marathon, he ran at a 5:30 mile pace, and he said it was terrible because he injured his hip at mile 9. 5:30 mile pace marathon. This is his first ever one. Anyway, we came to UConn at the same time, and whatever, he, I don't know where he is. He's like with the military or something now. So now getting back to a different gene. So that was ACE. Alpha actinin has held up over time. ACE hasn't really held up. Alpha actinin has held up. And it got famous. This isn't when it was identified. I have the article of when it was identified. This is about 5 years later, 2008. This is after Usain Bolt, right? And then they started testing Jamaicans for the alpha-actinin gene. And alpha-actinin holds actin in place at the Z-disk. Alpha-actinin holds actin in place. Now you have to adhere, you have to have proteins that adhere your filament, sliding filament theory, so that cross-bridge cycling can have some grip. There are 4 different types of these alpha-actinins, 2 and 3. Types 2 and 3 are in skeletal muscle. Type 2 doesn't really care what kind of muscle it's in. Alpha-actinin 2, type 1, type 2, whatever muscle, I'm happy to reside there. Alpha-actinin 3, this is a type 2 version. High threshold motor units. Big, strong, fast stuff. And so alpha-actinin 3 has a few genotypes. There are a few polymorphisms here. There's a strong version of it. And remember, this is holding, think of it like a tug of war. You're gonna do cross-bridge cycling, it's sort of a tug of war. The myosin sort of tugging on the actin. You have to have good grip. And alpha-actinin is sort of that grip. And if you're gonna do the tug of war wearing cleats, alpha-actinin RR genotype, that's the big, strong, that's the good one. There's a lot of Jamaicans have that. People all over the world have it, but it's more common in Jamaicans. The intermediate form, okay, that's like regular shoes. The XX, that's a nonsense snip, right? That's a premature stop code on there. That's like roller skates. Alpha-actinin isn't working there. And if you have this version, you're not gonna be a strength athlete. You're not gonna be a sprinter. It's just not gonna happen. And that's really held up over time. Now there may be exceptions, but that finding has really held up. But 2003, right, alpha-actinin genotype is associated with human elite athletic performance. They're looking at sprinting. 5 years later, people cared because Usain Bolt. Now, getting into which, this article you'll find, this article will be in the law, and it's a good one. It's July 2021, it's very recent, and it goes through all of the genes that have sort of held up. It talks about the ones that haven't, the ACE allele, stuff like that, the ACE gene. Those ones that are just a little bit too equivocal to list. But this article, this will be essentially the last slide. But there's a lot of genes that it talks about. They're highlighted in red, sorry, Liam. So all of the genes, those are the ones that it talks about. But there's good tables in here, there are good explanations. So if you're interested, I am not gonna test you on particular alleles. It does talk about the alpha-actinin 3, that RR genotype. But I'm not gonna test you on particular genes. Particular genes do pose a high risk toward diseases, conditions, right? A single gene can do that. But a single gene is unlikely to create an athlete. You need a constellation of tons of these things contributing, and we're nowhere near there. So the best thing you can do right now for a talent identification, to try to figure out what you're good at, is if you're Bowser's probably not a good endurance athlete. He's way in there in the back. Toad, that's a terrible gait. That's very inefficient. Way too many calories. Luigi's gonna be the best endurance athlete on the board. You can just go do the exercise. Go do a shuttle run. Go do, run for as long, do like a marathon or half marathon or something. Go run a mile, go run a 100-meter dash, go do some bench press. Are you good at it? That's a good indication that you're gonna be good at it. And it's free, right? Unless you have to pay for the marathon or something. So go test yourself with exertion, and that's a way better classification system than getting a cheek swab and saying, oh, here's the allele of this and whatever, this polymorphism, and I think you're probably gonna be pretty good at, X, Y, or Z. We're nowhere near ready for that. I think we'll probably get there at some point. By the time we get there, you can probably reprogram your genome anyway. So that's it. So that's exercise genomics. And we're gonna move on to exercise and brains next. And Liam, this is your last live lecture. All right. I will see people.